Hi everyone, good morning. My name is Biyanka Patel. A little background is that I'm a medical science liaison at Bavarian Nordic. Our company has a travel health vaccine portfolio as well as the vaccine for Mpox. And today I'll be talking about the Chikungunya virus disease state and then Vimkunya, which is our newly approved, FDA-approved Chikungunya vaccine recombinant. So a little overview. I'll be going over the virus and the transmission, the emergence and spread of the virus, and the pathogenesis, risk factors, disease burden, the impact of it, and infection, the patient's quality of life and how it affects them, the treatment and prevention of chikungunya, and then the overview of our vaccine-like particle technology for the vaccine and the vaccine trials, the 2 phase 3 trials itself. So the chikungunya virus is an arthropod-borne alphavirus with a single-stranded positive sense RNA genome. There are 4 different genotypes: Asian, West African, and then the East Central South African known as the ECSA, and then the Indian Ocean lineage, which roots from that ECSA genotype. Infections can lead to debilitating disease in individuals of all age groups and in both sexes. It typically presents with an acute febrile illness that's characterized by fever, fatigue, myalgia, and incapacitating joint pain. And many patients can develop chronic arthritis and arthralgia, which may persist for years. And due to its potential to cause a pandemic, chikungunya is listed as a priority pathogen for vaccine development by CEPI. So this virus is carried by 2 types of the Aedes mosquitoes, the Aedes aegypti on the left that you see here, and then Aedes albopictus pictured on the right. These species are commonly found in urban areas, and they preferentially bite humans. While chikungunya virus is primarily transmitted by Aedes aegypti mosquitoes, the ones you see on the left, in tropical and subtropical regions, there is a potential for a wider spread following the genetic adaptation of the albopictus mosquitoes, which usually thrive in the temperate regions. This map here shows the geographic distribution of both the Aedes mosquitoes. The virus is emerging as a global threat and has spread to the parts of the US as well, as you can see more on the western and southeast and northeastern parts. Now going into the spread of the emergence and spread of the disease. So the first outbreaks of chikungunya were recorded in Tanzania in 1952. The virus has since spread across the globe, causing large numbers of epidemics that have affected millions of people in Asia, the Indian subcontinent, Europe, Americas, and the Pacific Islands. And then during the 2005 and 2006 Reunion Island outbreak, there was a mutated strand of the virus that showed enhanced viral replication and transmission within the albopictus mosquitoes. And this is what accelerated the outbreak and spreading of this disease to a region that was non-endemic for chikungunya. Then in Europe, the first outbreak was in 2007 in Italy. And it was because of one man traveling from India. So it really sometimes, it just takes one person and one mosquito to cause an outbreak. And it's very, very highly unpredictable. And then 2010, 2013, and 2014 is when we observed outbreaks in France and saw the first outbreak in Americas. And then in 2014 and 2016, a total of about 3,900 cases were reported in the US from returning travelers. And by the end of December of 2015, there was a total of about 1 million cases with 71 deaths that were reported in the Americas And in 2016 alone, there was lab confirmed about 146,000 lab confirmed cases that were registered in the Americas. And then in Paraguay, from 2015 to now, there was the first autocanthonous case, and you could see the numbers that are listed below for that respective outbreak. And then in Brazil, also from 2015 to present, there has been about 175,000 cases from 2020 to 2022, which have been reported, and in about 2023, about 243,000 cases. So I'm going to go a little bit into the outbreak on the Reunion Island from 2000 that happened around 2005, 2006. This was a massive chikungunya virus outbreak on the Reunion Island. This island is, you can see it's a teeny tiny island circled here next to the island of Madagascar off the tip of Africa. And it infected about 1/3 of the island's population. And during this outbreak, the first reported cases of maternal to neonatal transmission had occurred. Death rates during this outbreak were about 34% higher than expected near the end of the outbreak. And acute symptoms in these patients were persistent, and 3 years after the acute infection, incapacitating polyarthralgia, myalgia, cutaneous lesions, and cognitive dysfunctions were present in 60% of these patients. And then now getting into the outbreak in Italy that occurred during 2007. So there was a man infected with the virus that had traveled from India to Italy, and he was the source of the first outbreak in Europe. So just one person that caused an outbreak. Infection was spread by the Albopictus mosquitoes, and then it led to the first outbreak in Europe in the Ravenna province of Italy. There were about 217 lab-confirmed cases over the course of the whole outbreak, and fever, asthenia, arthralgia were the most common symptoms seen. And 1/3 of the patients had persistent arthralgias 1 to 3 years after an acute infection, with functional impairment and relapses. And this, And this here, this slide here has the portrays the chikungunya virus outbreaks across the U.S. That occurred from 2014 to 2017. And before 2006 period, the virus was rarely identified in travelers. However, beginning in 2014, the cases were reported amongst U.S. travelers that were returning from those areas that were affected in the Americas. And there was local transmission that was identified in Florida, Texas, Puerto Rico, and the U.S. Virgin Islands. And then in 2015, the virus became a nationally notifiable disease. So the transmission that happened in Florida in 2014 was during the period of January, October, and it was a total of 272 cases. And in addition, there were 11 locally acquired cases that were identified, and the presence of these mosquito vectors provide the conditions for transmission of the virus in Florida. And this map here shows a geographic distribution of where cases have been reported, which consists of more than 100 countries and territories worldwide. This here is the estimated potential range of the Aedes aegypti and albopictus in US in 2017. These maps show areas where the mosquitoes are known or have been previously found. These maps don't, however, represent the risk for spread of disease. And then the past and current spread of the Aedes mosquitoes. So you'll see here in the purple and pink is where we see these mosquitoes today. However, there's factors that contribute to I have a next slide which I'll show in comparison. There's factors that contribute to the global expansion of this virus, and that includes climate change, because vectors, so mosquitoes, they're more able to expand towards temperate zones that were previously uninhabitable for them. And as a result of this climate change, some regions become more susceptible to outbreaks during climate change. Another factor that can contribute to global expansion of this virus is the mutation of the virus itself, so the viral adaptations, and the rapid growth of human populations and increase in the global interconnectivity with migrate, travel, trade, tourism. So this map here shows in pink and purple from the previous slide of where they are currently, but it's predicted for the mosquitoes to keep spreading throughout the Americas and Europe by 2050. So the biggest theme here is that the virus outbreaks are very highly unpredictable. And they're transmitted by mosquitoes that are located in many countries and regions. And the next outbreak could be anywhere in the world at any time, which gives us more of a reason to raise awareness on the preventative methods. And this virus has infected large amounts of people during an outbreak, with many people experiencing symptomatic illness. And the infection results in long-lasting immunity against the subsequent infections. So immunologically naive populations and travelers are the most at risk. Moving on to the pathogenesis, risk factors, and disease burden. Typical symptoms of the virus, of the being infected by chikungunya virus are fever, arthralgia, polyarthritis. You can see the picture here on the left with the arthritic-like symptoms with the swelling of someone's joints and their fingers, and then also backache, headache, rash. Arthralgia may be incapacitating, as I previously mentioned, and symptoms resolve within 2 weeks for most patients, but many do suffer from severe and debilitating long-term arthralgia and fatigue and often depression. Other long-term sequelae may include relapse of the rheumatologic symptoms, the persistent joint pain, and then neurological, emotional, and even dermatological disorders. The case fatality rate reported during the Reunion Island outbreak, which I went over a few slides back, was about 1 in 1,000 cases. However, mortality may be underreported due to the surveillance and reporting systems in the respective countries. So this slide here shows the acute phase leading to the chronic phase of when infection with chikungunya virus. So from day -10 to about -2, when you're getting the mosquito bite, day 0 is when there's the highest level of viremia that occurs within the serum. And then that's when you also, a few days after is when you also see the onset of symptoms. And then you do see the levels of IgMs and IgGs, so your own immune system that rises exponentially and sort of levels off after 2 weeks. And sometimes you could see the IgGs that are being persistent for months and years after. And diagnostic methods are also highlighted in this chart, and it tells you at what time periods which methods are appropriate. So you'll see in the red bars at the top where when you can do the rapid testing for PCR, the virus isolation, when the virus can be isolated, and then also when the serological test can be done. And chronic phase, I do want to highlight, is when a patient will see symptoms of arthralgias, or they may see symptoms of persistent arthralgias. Differential diagnoses include Zika virus and dengue fever. When you think dengue fever, you want to think bleeding thrombocytopenia. Zika virus, usually non-purulent conjunctivitis or transient Guillain-Barré syndrome. So the risk factors for the viruses, so the disease, are individuals who have not been previously exposed to the virus, such as travelers. Groups at risk for severe disease are newborns, older adults, and people with chronic diseases, such as those with diabetes and cardiovascular, neurologic, or respiratory disease. And this disease does affect quality of life and productivity. It has a significant negative impact on the quality of life and a persistent reduction in it up to 6 years after someone being infected. In one outbreak in Bangladesh, approximately 3 in 10 people with the disease lost more than 10 days of productivity because of severe arthralgias. Diving now into the treatment and prevention. So there isn't one specific treatment for this disease. It's more about the supportive care to alleviate the symptoms. Some options include acetaminophen, NSAIDs, narcotics, and for chronic joint symptoms, DMARDs, also known as disease-modifying anti-rheumatic drug therapy, are also suggested. It is important to avoid aspirin and steroids, and mild forms of exercise and PT may help with the recovery of the joint pain. Without an available vaccine, mosquito precautions are essential to reducing the risk of acquiring this infection. So CDC recommends the use of using EPA and picaridin, and the others listed there, and to also use permethrin to pre-treat your clothing and gear prior to perhaps going into an area where you may be at a higher risk, wearing long sleeve shirts and long pants, and choosing controlling the mosquitoes, being at a lower risk, and controlling your exposure to mosquitoes, such as choosing lodging with air conditioning or having screens with windows and doors, sleeping under a mosquito net, and disposing of standing water in order to avoid mosquito breeding. So all of this is just in summary, which I won't go over again. And now going into the virus-like particle platform and the Chikungunya clinical studies. So the Chikungunya virus, virus-like particle vaccine, it is a recombinant technology, and it comprises of three Chikungunya structural proteins. I didn't realize I didn't change the slide. I apologize. So the top here, you'll see the virus, and then at the bottom is the virus-like particle, which is what the vaccine is comprised of. There are three structural proteins Chikungunya structural proteins that the virus-like particle has. It's the capsid, envelope 1, and envelope 2, which help with mediating membrane fusion during infection and for binding to cells during the viral replication. Key properties of the virus-like particle vaccine that I want to highlight is that, as you can see here on the left, the structure is indistinguishable from the live virus. This VLP is non-replicating. The vaccine is adjuvanted with aluminum hydroxide to allow for a rapid immune response, and it's a single 40 microgram intramuscular route of administration dose. So a few other key features of the virus-like particles. They're highly immunogenic due to their similar size and structure to the actual virus. They do lack the viral nucleic acid, hence why they are non-infectious. And they exploit the immune system's ability to really recognize and mount a strong innate and adaptive immune responses against the viral structures. And their efficient activation of antigen-presenting cells and the subsequent T cell responses. So this was the assay, the luciferase assay, that was developed to evaluate the efficacy measures cross-neutralization. And so this was used to, this assay was based on the 80% reduction of the activity that followed with the varicocele infections with the chikungunya virus. And there was a conservative serum neutralizing antibody, the SNA threshold that was chosen by the agency for the phase 3 studies, which I'll get into. And uh, the non-human primates, they did receive the pooled sera from human participants that were vaccinated with the virus-like particle at various dilutions intravenously, and then they were challenged with the virus 24 hours later. In that logistic regression model, the titer of 50 resulted in 99.97% probability of protection against the viremia. Regulatory agencies then proposed a more conservative titer threshold of 100, just to be of 100 to be an acceptable surrogate endpoint, hence why we use that threshold in our clinical studies. These are the clinical programs that we have that have happened for the studies. And so the two pivotal phase 3 studies that I'll go into now, this is a design for both of them. So at the top, you'll see the adolescent and adults 12 to less than 65 years of age, and then adults greater than 65 years of age. So day 1 is when they received the vaccine, and then for day 22, you'll see the dashed red line that's when our primary immunogenicity endpoint was recorded. These subjects were followed up 6 months after, and so solicited, unsolicited, and serious adverse events were also reported and asked of them during the trials. So these are the primary and key secondary objectives in both the studies. For both of them, the geometric mean titers, also known as the GMTs, at day 22 versus the placebo were compared. And then the difference in the seroresponse rate versus the placebo at day 22 was also calculated. And then the third primary co-endpoint that they both shared was on the safety. Then for the first study, the log consistency of the geometric mean titers at day 22 was also recorded. For both, the key secondary endpoints was the sera rates. For the first study, it was also day 8, but then for both, it was day 15 and then day 183, and that was compared to the placebo. This here represents the demographic characteristics in the adolescents and the adults studies, and it's basically saying that the baseline characteristics in the individuals were similar. And this is for those in the greater than or 65 years of age. So here you'll see that at day 22, the seroresponse rate difference between vaccine and placebo in the first study was 96.6%. And then the vaccine group geometric mean titers was significantly higher than that of the placebo. And the pairwise log comparison of the SNA response to the virus in adults did demonstrate equivalence, hence why all the co-primary endpoints were met. As well as all key secondary endpoints were met as well, with comparison of day 8 and then the following days afterwards, up until 6 months. You'll see a similar curvature on both the graphs for the second study in the older adults, because also the co-primary endpoints were met. However, the rates were a little different. So at day 22, the rate difference between vaccine and placebo group was 86.2, and then the geometric mean titers was significantly higher than that for the placebo as well. And all key secondary endpoints, hence all key secondary endpoints were also met. So all co-primary endpoints were achieved. This shows more of deeper data in terms of numbers. So individuals, so the vaccine seroresponse rate showed statistical superiority to placebo, and beneath it you could see between the two trials what the our seroresponse rates were. The geometric mean titers at day 22 was statistically significantly higher than that of placebo, and then for the first study, there was equivalent in pairwise equivalence seen between the three consecutively manufactured vaccine lots. And similar, the key secondary endpoints were achieved in the pivotal studies as well. And the seroresponse rates for both studies showed a statistical superiority to that seen in the placebo. Is that the time? 5 minutes. Okay, I'll be really quick. So this year, the vaccine showed an acceptable safety profile in individuals. In the first study, the rate, fatigue, headache, and myalgia were the three systemic solicited events that were observed. And then the solicited systemic events first week after the administration was injection site pain, followed by redness, and then swelling. And you can see the different bars here on the graphs with their percentages of who observed what in terms of rates. And then one participant had a grade 3, had a severe treatment-related unsolicited adverse event of dehydration, which did resolve without medical intervention. However, it was possibly related to that subject having a prior medical history of seeing black spots in the right eye 1 month before study. And then also in the older adults population, the solicited local AEs 1 week after the administration of the vaccine were injection site pain, and then very little in the placebo group you can see, but that's not relevant because it was in the placebo group, but it was injection site redness. And then for solicited systemic AEs 1 week after, myalgia, fatigue, and headache were the top 3 that were reported. For unsolicited AEs, fatigue and myalgia were the only treatment-related unsolicited AEs from the previous slide that I mentioned that occurred in more than one participant, and no AE was considered treatment-related. So to summarize, this is my last slide, I promise. Single-dose vaccine based on the virus-like particle technology is what this Chikungunya vaccine is, and it's indicated to prevent disease caused by the virus in those that are 12 years of age and older. Oh, I'm sorry, I didn't skip. It is approved by the FDA on February 14th as Chikungunya. The phase 3 trials demonstrated immune response in individuals 12 years of age and older. There is a durable and boostable immune response in the phase 2 trial. We did observe acceptable safety profile. And the most common solicited local adverse reaction for patients in the first study was injection site pain. And the most common solicited were fatigue, headache, and myalgia, as I mentioned earlier. And then for in the older adults population, those that were 65 years and older, the most common solicited local event was injection site pain, and the most solicited systemic was myalgia and fatigue. Also, I know we don't have time for questions, but if anyone has any comments or questions, please feel free to email me at this email or even send in a medical information request at that email. Thank you. No, it's not the actual virus. In fact, there are no actual components of the virus in this virus-like particle.
So what is the basis of that virus-like particle?
Yeah, they're proteins. They're structural proteins. And they are assembled in a way that resemble the proteins found on the virus. So this right here, sorry, I need to go back. I want to show that one slide real quick with the actual virus and the VLP right here. So this structure resembles it, but it doesn't have any actual components of the virus. It's not live, it's not attenuated.
Sure.
It's created with proteins. It doesn't have any genomic material, hence why it's non-replicating. It's created from proteins.
And it carries the proteins, envelope and structural proteins of all the 3 serotypes?
Sorry, what was the question?
Strains, there are three strains, you said there are three strains of
Yeah.
chikungunya, right? Does it
No.
carry all the three strains, surface proteins?
Yeah, we can defer to Alejandra. She's our specialist.
Although chikungunya has 4 different genotypes, it behaves as one, thank you, as one serotype. So we believe that the assay already shows that we have cross protection, and we believe that the vaccine protects for all 4 serotypes because genotypes, because they behave as one single serotype.
So the proteins that are on the VLP are based on one serotype's
Yes.
amino acid structures. And also, how long did you follow the subjects for potential antibody-dependent enhancement? Was that part of the study? Or?
Yeah, so for this first part of the study that we have the results publicly available, we followed them for 6 months for safety reasons, but we still have ongoing trials with the same population, but we don't have the results available yet, but
Okay.
we are following them.
Thank you.
Thank you.
All right, we don't have more time, sorry. So our next speaker is
Oh, sorry.